Hello and welcome to another episode of Odds and Ends Season 2. In this episode, we'll talk about the history of the Pangenome and its applications to agriculture and botanical study. The first known instance of the Pangenome was started with an attempt to make an effective vaccine for Streptococcus agalactiae. During this time, the prevailing model was that the genes of a species were fixed, and the only differences are differences between versions of those genes called alleles. More on that later. Dr. Mayone produced the idea of using the genome sequence of Streptococcus agalactiae, from here on out called SA for short, to find out what proteins promote the creation of antibodies so that an effective vaccine could be created. What was found in the experiment was that the different strains of SA did not have an allelic counterpart in other strains. This happened with half the genes, and no matter how many times they attempted to make this genome fit within the current paradigm, it just did not work. Further genetic testing found that 80% of the genes were utilized in cell division and completing the life cycle of that bacteria, and followed the fixed genes varying alleles model. The other 20% did not, as the other 20% of that genome found within all samples of that species they had access to not only contained different alleles but different genes altogether. From there, it's concluded that the SA bacteria had two different genomes: the core genome, which maintains all the genes needed to have proper functioning within the organism, and the pangenome, which have genes that are free to vary, both in number and type. It was later found that eukaryotic species, which are to say plants, animals, protozoa, etc., also fall under this category of having both a core genome and a pangenome. The first species discovered to have such a system was baker's yeast and two candida species. In later periods, common crop plants were also found to have pangenome. In a study where 7 populations of soybeans were studied and genetically analyzed, it was found that 58% of the genes were unique to 2 out of the 7 populations, and 42% were unique to only 1 of the populations, which is to say that if you look at all the genes available, 58% of the genetic differences between groups of 2 populations differ incredibly from other groups of population. And there's a 42% difference between the number of genes and the type of genes from one population to another. In rice, it was found that 38% of the genes within that species were free to vary and could be considered part of the pangenome. For corn, the pangenome was found to be 61% of the genome of the entire species. In cabbage, it was found to be 19% of the entire species. Sources for these variable genes needed to be accounted for. And after reviewing different papers and looking over different experiments, 6 independent sources of this genetic information was found. The first was interspecific hybridization, which was first discovered by Dr. Edgar Anderson in 1949. It was found that in Pacific families, distinct species of plants can crossbreed with each other, allowing new genes to be integrated into other species, becoming part of their genome. It is also found in certain finch species and pisli. Next would be whole genome duplications, where the entire genome is duplicated, with that duplicated genome mutating until it resembles nothing identical to the original genome, becoming new traits entirely. Wheat is one such example. Another source would be segmental duplication. In humans, this is the cause of a great many genetic disorders. This is where a portion of a genome larger than a single gene gets duplicated. The primary source of pangenome genes within beer yeast is this process. The next source would be horizontal gene transfer. This is where genes from another species get incorporated into a host species. Sweet potato is a good example. This species has a close relationship with a species of bacteria called Agrobacterium. The symbiotic relationship is very interesting. The bacteria incorporates its own DNA into the sweet potato, causing it to produce those large tuberous roots, but it also incorporates genes for disease resistance as well into its genome. That bacterial DNA remains conserved even in populations without the agrobacterium. So a good chunk of the DNA within sweet potato originates from a bacteria enough. The last method we'll cover is single gene duplication. This is where a single gene gets duplicated and becomes free to vary through mutation. Rice is a good example, as based on current genetic evidence, most of the unique genes are formed through this process. Indeed, right now, people are even finding that this is happening in real time through observations of the genome within rice. Most genes in the pangenome seem to be used to develop disease resistance or abiotic stress resistance, such as drought resistance. Now on to the applications of new discovery. The pangenome has been used extensively for different crops and species, using the concept of presence-absence variation, where a gene is either present or absent within a cultivar or population. Various forms of research in plant genome have found that the pangenome is a source of different traits such as flowering time, disease resistance, and agronomic traits, like environmental stress response. Most researchers right now are looking into the pangenome of various cultivars and populations of different crops to try to find novel genes so that they can incorporate genes that are favorable from one plant into another plant that also has different favorable genes. People are also using this to look at the evolution of different species. By looking at the different amounts of variation within the pangenome, they're able to find out that plant genomes are highly dynamic, with gene size varying between time and space. This technology is currently being used to study the different lineages of different plants and figure out at which point the species diverge. The existence of the extensive corn plant pangenome showcases that there is still an extensive amount of genetic information to draw from to further improve the crop, and that if you want to breed corn, combining different plants that have not just different alleles but different genes is very much possible and desirable. Due to the greater complexity of a pangenome versus a typical corn genome model, it's more difficult to have access to it through statistical means. As a consequence, machine learning has been used extensively. New algorithms called deep learning models are being used extensively to develop new models for the genetic variation within the pangenome and the genetic structure of a pan. A spin-off of this technology is to use this technology to find out how much off-target mutations happen when you use CRISPR-Cas9. These deep learning models are still in their infancy, but it is speculated that once they're ready, we could be able to understand the full complexity of the plant pangenome, rather than small parts of it here and there. And that about covers everything. Thank you for watching.